Hi, I'm Bob Pignolo, and I'm visiting today from Mayo Clinic in Rochester, Minnesota. I'm the Robert and Marlene Kogod Professor of Geriatric Medicine and Gerontology. So the title of my talk is Old Bones and Second Skeletons, and really that title reflects the two areas in bone biology that I've devoted my career to. On the one hand, old bones, osteoporosis, fractures, bone loss in the normal skeleton. On the other hand, second skeletons refers to heterotopic ossification, which is bone outside the normal skeleton. It's normal bone, but it's in the wrong place. And to me, the idea of merging these two areas was intriguing because initially I thought if one could understand how bone is made outside the normal skeleton, this is real bona fide bone, normal bone, outside the normal skeleton, even in older individuals, maybe that could tell you something about targeting age-related bone loss. The fact of the matter is it worked the other way around. So my study of age-related bone loss actually informed at least one target, a therapeutic target for heterotopic ossification. So I'm going to focus on what links the two processes that seem very disparate. And the theme here is cellular senescence. So cellular senescence is a process by which cells, through various inducers, undergo a state of proliferative arrest and assume certain characteristics, including extreme apoptosis resistance and the formation of what we call an inflammatory secretome. And so these are factors that are secreted by senescent cells that have deleterious effects both locally and even systematically. And so cellular senescence links both age-related osteoporosis and heterotopic bone formation, and they contribute to both processes in very interesting ways. So as I mentioned, cellular senescence is a mechanism that is at play in both age-related bone loss and in heterotopic ossification. In the former case, the case of age-related osteoporosis, cellular senescence seems to play several different roles. The first role is really changing the stem cell function in bone as we age. So normally, stem cells in bone contribute to bone formation by differentiating to bone-forming cells called osteoblasts. Well, with aging, there's a lineage switching that occurs in these precursor cells, and instead of making bone-forming cells, the pathways change to adipogenesis, so fat is formed instead of bone. This is not a passive process. It's really a lineage switching, if you will. Also, there are other cell types in bone that become senescence, which really causes an imbalance in bone formation versus bone resorption. Normally, bone formation is coupled to bone resorption, but with aging, it's just uncoupling of that process, which mediates bone loss that we normally experience with age that predisposes to fractures. In the case of heterotopic ossification, as a result of injury and other insults, especially to soft tissues like skeletal muscle, cellular senescence is a process that occurs as a direct result of the injury. So you have an accumulation of senescent cells. And senescent cells in this context make factors which both promote tissue reprogramming, so that instead of healing the injury, instead of a regenerated muscle, there's formation of bone instead of new muscle. And that's related to both reprogramming factors that are made by senescent cells, but also, at least in the case of one particular genetic condition of heterotopic ossification, other factors which promote lesion formation that ultimately causes the formation of extra bone outside the normal skeleton.